Welcome, one and all, to another episode of Soil and Crop Sciences Seminar, the greatest show on earth. At least that's what I'm told. Today, we've got a pair of students who are eager to tell you about their project accomplishments. Dr. Smith, would you like to introduce our first speaker?
I would love to. All right.
Much enthusiasm.
So I'm coming to you live from soil morphology class. You can see this is the class that Nicole is usually teaching during seminar. So I am so excited to present Nicole and introduce you to her. I met Nicole when she was an undergraduate and she instantly impressed me. One of my first memories is when she came to my office to discuss some of the results she had gotten from an undergrad research project and was coming up with her own hypotheses for explaining those results. But then she reminded me today that actually our first encounter was geeking out over dorky soil science shirts and stickers, which is also very true for both of us. So she got a double major in soils and horticulture 2 years ago, and then she will be graduating probably this summer with her master's. And another thing I wanted to point out is that in the past 2 years, both her and Anna Lee have had analyzed over 500 samples for their master's project in several different indicators. So that's a lot of work. And so she's been working hard and also teaching this class throughout her entire master's degree. So I'm very proud of her and very excited to hear what she has to say today. So take it away, Nicole.
So can everybody hear me?
Yes.
All right. And can we see my screen?
It is visible.
Okay. So, hi, I'm Nicole Shigley. Like Dr. Peyton Smith said, I am a second-year master's student here at Texas A&M University, and I will be telling you a little bit today about my master's project with how tillage impacts soil carbon cycling. And again, I want to give a little shout out to my lab partner, Anna Lee. She's giving a presentation and seminar later on this semester, and I thank her for helping me with analysis as we work on this project together, and our two departmental advisors, Dr. Julie Howe and Dr. Peyton Smith. And this picture that I have at the front was pre-COVID field trip out to our site where I got to talk about our research some in person. So I wanted to start off with, why is my study important? A lot of the physical and the chemical aspects of how tillage impacts soil are pretty well studied. We know that tillage decreases bulk density and it homogenizes soil above this till line. We also know that tillage increases aeration and it breaks down soil structure, which exposes physically protected nutrients, and this can increase the nutrient cycling in the soil. But the biological impacts based on tillage are a little bit less understood. But it's really important to understand these because the microbes in the soil are these key drivers to nutrient cycling for productive crop production. And as these studies in microbes start to get more advanced in the agricultural field, a lot of them still just focus on the surface soil because this is where a lot of the activity is thought to take place. So starting with my experimental approach, I was lucky enough to get to use some of Dr. Howe's plots that she took over when she came here. These are long-term sites that have been in the same management practice for over 35 years. So everything I'm studying is the long-term effects of tillage on cropping systems. And we are located at the Texas A&M Research Farm near the Brazos River. On my Texas figure to the left, I have Brazos County highlighted in hot pink. And on the right, the figure shows an aerial view of our fields. And the two fields that we're going to be talking about today are highlighted with some hot pink boxes. So we are in Brazos County, and our overall field design is a randomized complete block design. These fields are in a crop rotation of sorghum, wheat, and soybean. And for the rest of this project, I'm going to be kind of splitting it into two different projects. We have a seasonal study and a depth study. So in our seasonal study, we took 8 composite soil cores 4 times throughout the growing season. We're going to be looking at no-till and conventional till, but we're just going to focus on this top 0 to 5 centimeters for the seasonal study. These pictures here is my lab partner, Anna Lee, taking a soil sample and a picture of me fertilizing our sorghum crop field. And then for our depth study, we took one sampling of deep cores with a Giddings probe, and these were taken right after our winter wheat harvest. Again, we're going to look at no-till and conventional till, but our deep cores got split into several different depth increments, and these are 0 to 5, 5 to 10, 10 to 15, 15 to 30, 30 to 60, and 60 to 100 centimeters. And the pictures I have here, the picture on the left is a picture of one of our cores taken out of the field, and the picture on the right is me helping with our wheat harvest. So going into our lab analysis, so we are in a Westwood silt loam. It's an inceptisol, which means that it is a younger, less weathered soil. It still has a lot of base cations in it. And for my soil specifically, we have a lot of free carbonates in our soil. And so in order to get our organic carbon measurement, we need to take out that inorganic carbon component. And so I did inorganic carbon with the pressure calcimeter, and you can see a picture of my lab setup for that. And we subtracted that from our total carbon that we did with total combustion, as seen on our picture on the left. And this together gave us our organic carbon percent. The other measurement that we're going to look at is our active carbon. So active carbon today we're going to be looking at POXY, which is permanganate oxidizable carbon. It's a colorimetric method, and it tells us the easily oxidizable organic carbon portion. And this relates to the more readily available carbon for microbes. So the carbon pools there's a bunch of different carbon pools, but we're going to focus on these two for today. A little bit of a picture to kind of break down and understand the carbon components a little bit more. We have total carbon in the soil, which is the organic and inorganic components. When you take that inorganic part out, you're left with organic carbon. And then an even smaller portion of that organic carbon is the active carbon that we'll be talking about as well, the easily oxidizable stuff that the microbes are more readily able to use. Moving on to our biological measurement, we use enzyme activity as a measure of our microbial activity. So we used a fluorescent method. We chose this method as it is more sensitive to management changes than the older colorimetric method. And this is kind of our lab setup for it. And the picture on the top left is where we have our buffer and our blender where we make our soil slurries. The picture below that is kind of a full run that I would do for the day. And the picture on the right is me setting up and prepping and putting those substrates into the trays. All of our substrates for this fluorescent method are linked to fluorescent compounds. When the microbes start eating those substrates, it breaks the bonds, and this makes the sample fluoresce. The more that it fluoresces, the more activity that is happening in the soil. So we're going to focus on three components that cycle carbon, nitrogen, and phosphorus in the soil. So first we will have beta-glucosidase, which I will refer to as beta-gluc, and this cycles carbon. We have phosphatase, which I will refer to as phos, and this cycles phosphorus in the soil. And then we have chitinase or nagase, which I will refer to as nag, and this cycles nitrogen in the soil. So we will start off into our data analysis. We'll start with the seasonal part of the study. Again, this was during the sorghum growing season. And we're going to start off with looking at our two different carbon pools. And we saw that the tillage reduces carbon throughout the growing season. So this first graph here is organic carbon. We have our sampling collecting points at the bottom based on the growth stage. And then we have organic carbon percent going across the y-axis. This figure shows that there is an impact of tillage across the entire growing season. No-till is higher than conventional till for our organic carbon pools for the whole season. And again, we see a similar pattern in our active carbon. Again, we have our sample collection points based on growth stage across the x-axis, and our epoxy in milligram per kilogram of soil across the y-axis. And you see here another significant impact of tillage across the whole growing season, with no-till having more active carbon than the conventional till. Next, we wanted to look a little bit into our microbial activity. So starting off with beta-gluc, this graph is set up where we have our tillage effects on the left in the pink box plots, and we have our growth stage effects in the brown box plots to the right. And then across our y-axis, we have our beta-gluc activity levels. And so here for our tillage effects, there is an overall tillage effect where no-till is higher than conventional till. And for our growth stage effects, you can see that we have the highest beta-gluc activity in our germinated stage, which makes sense. The plants are growing, they need all that carbon. And then we have our lowest in the harvest section. When the plants are done growing, they shouldn't really need any more carbon assimilation. Moving on to NAG, again, we saw both an effect of tillage and growth stage. So on the left, we have our tillage effect, and on the right, we have our growth stage effect again, with our NAG activity across the y-axis. So here we have effective tillage, where no-till is still higher than conventional till. And in our growth stage, we actually have higher levels throughout the beginning in just planted and germinated stages. And our lowest is still again in that harvested time. And then our last enzyme that we will look at for the growing season is phosphatase. So again, we have an impact of tillage and growth stage here as well. The tillage impact is on the left in pink, growth stage is on the right in brown, and then we have our phosphatase activity levels across the y-axis. And here we see an impact of tillage where no-till is higher than the conventional till. And then for the growth stage, again, we see similar effects as we did with our beta-gluc, where the germinated had the highest phosphatase activity when the plants are growing, and harvest had the lowest when the plants are done growing. So what are these key take-home messages that we got from our seasonal study? We saw that the tillage decreases these carbon pools at that surface depth across the entire growing season. We also saw that the tillage decreases our enzyme activity across the entire growing season. And so our results are suggesting that maybe these higher organic and active carbon pools are what's driving that increase in extracellular enzyme activity. And then we'll go ahead and move on into our depth data analysis. So this is the samples, the deep cores that we took right after our winter wheat harvest. Again, starting with our carbon pools, we'll start with our organic carbon. I set these graphs up a little bit different. I stuck with the box plots, but here I have organic carbon across the x-axis, and I did depths across the y-axis to better reflect a soil profile looking down. Here we do not have a tillage effect throughout the entire profile, whereas in the seasonal, we saw the effect across the whole growing season. And we saw that does not reflect down the soil profile. We only have a significant tillage effect at that 0 to 5 centimeters in our organic carbon pools. And then moving on, looking at our active carbon, again, we have epoxy in milligrams per kilogram of soil across our x-axis, and we have depth going down our y-axis to reflect the soil profile. Here, there is a little bit interesting difference. We have 2 points where tillage does make an impact. Again, at that 0 to 5 centimeters, where no-till is higher with active carbon than the conventional till. But then again, we also see another significant difference at the 10 to 15 centimeter mark, where conventional till actually has more active carbon than our no-till. And this is interesting to note. It's right at our till line at about 15 centimeters. And then moving on and looking into our microbial activity, again, we did not see an effective tillage through the depth of the profile. There is one significant effect of depth or effective tillage at the 0 to 5 depth in phosphatase, but there is no other effective tillage anywhere else throughout this, throughout the enzymes. There are, however, some interesting trends that we saw. If you look at the depth 15 to 30 across all 3 enzymes, there's a big variance in our conventional till, and this is right under our till line. And then also at our 10 to 15 centimeters in our no-till for phosphatase, phosphorus is an immobile nutrient, so I assume that wide variance in enzyme activity has to do with where our phosphorus fertilizer is placed in our soil. So what did we learn from our to depth studies? We learned that surface soils are more sensitive to tillage. The tillage only affected our carbon pools at that top 5 centimeters. And then for our enzymes, it only affects the phosphatase enzyme at that top 5 centimeters. So kind of wrapping everything together and bringing together a whole message, we believe that the residues that are left by these no-tills are building up those carbon pools in that top 5 centimeters, and it's leading to a more biologically active soil throughout the growing season. So like Dr. Smith said, we've got lots and lots of data, lots and lots of samples. So this is talking about future work and what Annalie and I will continue doing in our study. So for our seasonal study, we will have 2 years of seasonal data. We will add on alpha-gluc and CBH enzymes. These are both also carbon cycling enzymes. We will also have nitrate and ammonium data add. And for everything with our seasonal 2-year study, we have a second depth of 5 to 10 centimeters. And something interesting with our seasonal plots is that we have 2 sets of fields that have the same crop rotation planted in even and odd years, so that after the 2 years, we will have both 2 years of soybean data and 2 years of sorghum data to look at. Today, we only looked at sorghum just to compare the same exact plots for seasonal and to depth study. And then going on into our future works for our depth study, again, we will also have alpha-gluc and CBH enzymes as well added to that. We will also have nitrate and ammonium to look at that, but something a little bit more specific to the depth study is we will get to compare a monoculture profile to a crop rotational profile. I'm really interested and excited to see the differences between crop intensity as well. So with that, I will leave you with kind of our take-home messages from each of our sections. The seasonal sorghum, we saw that long-term tillage depletes our carbon and microbial activity at the surface throughout the growing season. And for winter, we saw that those surface soils are more sensitive to the tillage practices. And then I wanted to end on a fun picture. This is Annalie and I on our last soil collection. I think if you add in our winter samples and stuff too, we have closer to over 700 samples, which is quite a lot to do, especially when you have to do redos. So with that, if you want to contact me any further after this, you can contact me at nshigley@tamu.edu, or feel free to join me on social media. I have an Instagram set up at Hola My Soils, where I am kind of exploring soils through my teaching, through my master's program, and just sharing my knowledge and trying to get the word of soil out there in the public. With that, I'll take any questions.
Very good, very interesting work. I'm sure there's going to be a lot of questions. Questions?
I have one. So the importance of the soil microbiomes becoming more and more, noticed as an important factor in plant productivity. Did you see a difference in the yields or the overall plant performance from your no-till and till? I mean, I'm guessing you probably use the same fertilizer techniques, but
Yes.
just curious if you had any insights on that.
So our field is a lot more complex than just looking at tillage. We also have different rates of fertilization as well. Annalie will talk a little bit more later in the semester about the differences of the nitrogen rates that we add. But yes, so we are seeing a little bit, I think the differences are more with tillage, but tillage also influences the microbes. Everything is so interacted together. I am interested in seeing if there are differences in the microbial populations. I think that would bring the research a little bit further too, instead of just activity on the certain nutrients. But I do think that there is a difference between the microbes. We see that there's more activity in that surface soil, but with the tillage, you could see right under that till line, we have an increase of activity there too. With our crops, our sorghum is doing great. Soybean is kind of struggling. I'm not sure if that's just our climate or if that has something to do with our soil.
Cool. Yeah, thank you.
Oops.
Next,
Hi,
Hello.
Nicole.
go
Oh,
ahead.
sorry.
Go ahead, jump in there, Megan.
I was just wondering, you were looking at sorghum and you're looking at wheat, and one of those is a C3 plant and one is a C4 plant. How does that impact, your particular study that you're looking at regarding the carbon?
So we haven't looked at impacts based off of the C3 and C4 plants, but we have noticed when we looked at some prelim data that there is crop influence, even from our soybean to our sorghum, both of our summer crops. We have to separate it by crops before we look at data. So I do think that there is a big influence on crops as well.
Can I jump in just a second and say that all of these treatments, it's the same 3 crops in the same fields every year, or every 2 years, I should say.
Yeah, we have our sorghum, and then wheat follows that summer, and then soybean, fallow, and it keeps rotating constantly.
All right, thank you.
Hello, Nicole. This is Yojun. I want to ask you a question more related to soil mineralogy instead of carbon, right? So since you have analyzed a lot of samples now, so what's the calcite content range in your soils? Didn't they show any trends in terms of depth or spatial natural differences in your field?
Are you asking if there's differences over time or just different spatially?
The different spatially, firstly.
For
What's the general range of your calcite in your soils?
So when we see our total carbon numbers, we have like the max amount of total carbon in our field I've seen is about 2%
Okay.
for our 0 to 5 centimeters, and it's usually a little bit lower than that for the 5 to 10 centimeter range. And then when we take out our inorganic carbon, all of our field has less than 1% or organic carbon. It's pretty low.
Thank you.
You're welcome.
More questions?
Hi, I have a quick question. So, can you hear me?
Oh, yeah.
Okay.
Can you talk a little bit louder, or I'll turn my volume up too.
Okay, so I have a quick question on the nitrogen pools. So, did you have any study going on or any plan to look at the different nitrogen pools on the side by side with the carbon pools? Because maybe the carbon pools are working better in the no-till, but how it's going on in terms of nitrogen pools, and also correlating them with the yield would be a nice presentation. So do you have anything about the nitrogen pools?
Yes, so we're going to be looking at the nitrate and the ammonium. So my study specifically is focusing on just the high nitrogen fertilization rate plots. Annalie's study later on is focusing on a high rate and a low rate of nitrogen fertilizer and how that influences the carbon cycles. So that might relate a little bit more to her project. I will be looking at nitrate and ammonium as well, though, later on.
Yeah, that's good. Thank you.
More questions?
I have one question.
Go.
Why is the microbial activity is lower in the flowering stage? Is that related to irrigation or something else?
Could you repeat that one more time?
So microbial activity is lower in the flowering stage.
Oh, in the flowering stage.
Yeah.
Yes, go back. Oh, we're looking at this one.
Yeah, here. Is that related with irrigation or something else?
So our plots are not irrigated, it's just rain fed.
Sorghum also. Sorghum and
Yes, all of our fields.
okay, thank you.
You're welcome.
Other questions? Yeah, I'd like to just follow up on Mustafa's question, Nicole.
Okay.
Why do you think there is that decline in activity?
Okay, let me go back to that. So I'll just stick with this one. So when those plants are just kind of growing, it makes sense to me that that's the increase in activity. That's when they're growing the most. Flowering now switches from a vegetative to
What are you referring to as growing the most? The microbes?
Oh, no, the plant. So
Okay.
sorry. Yeah, let me clarify that. So germination, the plants are growing the most. So that's when they're going to need a lot of nutrients constantly for growing and the vegetative production. And then in that flowering section, they change from high inputs on the growing into the vegetative section, or the vegetative to reproductive. And then we start to dry out and get ready for harvest. So that's where I see that decrease in.
So your hypothesis is the plant is driving the microbial activity?
Yes, I do think that the plant and microbes have a big influence together. And like I said before, even just looking at our different crops, there is a cropping influence, and we have to separate our crops when we look at our enzymes.
Is it possible, I think Mustafa was mentioning that there might be more soil moisture at the beginning of the season, or maybe there's more carbon in the soil at the beginning of the season from the previous season's waste matter?
Right, and we see here also higher carbon at the beginning, and then it kind of drops when our plants start growing. I do think that will input it, depending also how much residues left over from that crop before. I think crop has a lot of input into it, yes.
Also just gonna add that the enzymes that she presented, she actually normalized it by organic carbon in the soil as well, just to make up for that. So you're not seeing an effect of, a higher substrate.
Okay. Any other questions? Going once? Going twice? Thanks, Nicole.
Thank you.
Dr. Vaughn.